Welcome back to the deep dive. Today, we are shifting gears a little.
We are.
We're going into full-on, high-stakes study session mode. So if you were a student sweating over exams on regulatory science, pharmacology, or toxicology...
Or maybe you're in the industry.
Right. Maybe your boss just dropped a 30-page PDF on your desk and said, figure this out. This deep dive is for you. We are tackling a document that is the absolute bedrock for gene therapy development.
It is indeed. But let's be honest, it's also the kind of document that can cure insomnia if you try to read it cold.
Exactly. We are decoding the ICH S12 guideline on non-clinical biodistribution considerations for gene therapy products.
Yeah.
Now, I know regulatory guideline sounds incredibly dry.
It does.
But I want to reframe this. Imagine you spend 5 years developing a cure for blindness. You have the vector, you have the payload, you've cured the mice in your prelim study, you're ready for humans. And then the regulator asks,
but did any of that virus end up in the ovaries?
Precisely. If you don't have that answer, your program is dead in the water. That's the power of ICH S12.
That's the perfect way to frame it. This is not just paperwork. This guideline, adopted in March 2023, is essentially the GPS tracking manual for gene therapy. It answers those fundamental questions. Where does the therapy go in the body? How long does it stay there? And how does it get out?
And before S12, this is a bit of a Wild West, wasn't it?
It was. You had different regions, the US, Europe, Japan, and they were sometimes asking for slightly different things. You might run a study that satisfied the FDA, but, left the EMA asking for more data.
So S12 harmonizes all that.
It does. It's like getting a universal passport for your data.
Okay, so let's start at square one. If I'm sitting in an exam hall and I get asked to define biodistribution, or BD, in the context of this guideline, what's the gold standard answer? Because I feel like I could just say where the drug goes, but that won't get me full marks.
No, where the drug goes is the conversational answer. For the exam, you need to hit 3 specific keywords. Biodistribution is the in vivo, distribution, persistence, and clearance of a gene therapy product.
Distribution, persistence, and clearance.
Okay. Right, and you have to specify that this is happening at the site of administration and in both target and non-target tissues, including biofluids.
Okay, let's pause there. Distribution, persistence, clearance. Distribution makes sense, that's the spread. But persistence and clearance sound like two sides of the same coin.
Ah, that's a great question.
I mean, if it persists, it hasn't cleared. Why do regulators need us to measure both?
They're related, but they tell you different things about safety. Clearance is about the rate of removal. How fast is the body scrubbing this out? Persistence is about the duration of what stays behind.
I see.
You need to know if the genetic material is just hanging out in a tissue, like the liver, for weeks or months or even years. Persistence drives those long-term toxicity questions.
Got it. So if it clears from the blood in an hour, but persists in the liver for a year, that is a massive safety signal.
Precisely. And remember, we are primarily tracking the genetic material itself, the DNA or RNA, not necessarily the protein it produces.
Right. Now, before we get into the weeds of study design, we need to draw a circle around what this guideline actually covers. Because I feel like this is a classic exam trap.
Oh, for sure.
I can just see the multiple choice question now. Which of the following is regulated by ICH S12? So what counts as a gene therapy product here?
It is pretty broad. It includes products that mediate their effect by the expression of transfer genetic materials. So think purified nucleic acids like plasmids or RNA.
Okay.
It covers genetically modified viruses, bacteria. It covers ex vivo genetically modified human cells. And it even covers gene editing products.
So if I'm using CRISPR, S12 applies. So what is out? If I'm writing a test to trick students, what are the distractors?
2 big exclusions to remember. First, prophylactic vaccines are out of scope.
Okay.
So if you're making a vaccine to prevent an infectious disease, even if it uses similar tech, S12 is not your rulebook. Second, chemically synthesized oligonucleotides are also out, with a few caveats.
And there's one more exclusion that actually surprised me when I read it. Shedding.
Yes, and this is where people get confused. Biodistribution and shedding, they sound like they should be the same thing.
Right. You're tracking the movement of the drug.
But they are totally different concepts for the regulator.
Break that down.
Biodistribution is what happens inside the body. It's the internal map. Shedding is the release of the product outside the body. So through excreta, secreta, or through the skin. Shedding is about environmental safety, the risk to other people like family or nurses. Biodistribution is about the safety of the patient. S12 is strictly about the internal GPS.
Okay, that is the vital distinction, internal versus external. So we know what it is and who it's for, But let's talk about the why. Why do regulators care so much if a little bit of the vector ends up in the liver if we're targeting the eye? It feels like micromanagement.
It's not micromanagement, it's context. Your biodistribution data is the lens through which you interpret everything else. Imagine you run a toxicology study and you see liver damage. If your BD data shows the drug never went to the liver, then that liver damage probably isn't a direct effect of your drug. Maybe it's secondary, an immune reaction. But if your data shows the liver is full of your vector,
Then you have a direct correlation.
you do. It also helps you design the first-in-human clinical trials. It tells you which organs to monitor in your first patients.
So it's the map that tells the safety monitoring committee where to look. Now, let's move into the core concepts. When do these studies need to happen? Can I run them alongside my phase 1 trial to save a little time?
Absolutely not. That is a critical failure. These studies must be completed before you initiate clinical trials. The whole point is to inform the risk assessment before you treat a single human.
Makes sense.
If you don't know where the drug goes, you can't ethically dose a person.
But this brings up a tension, doesn't it? We want tons of data to be safe, but we also have an ethical obligation not to use thousands of animals.
Right. And the guideline is very conscious of the 3Rs principles: reduce, refine, replace. It's a balancing act. You need enough animals to get valid data, but you're also mandated to avoid unnecessary use.
So you have to get it right the first time.
Exactly. This is why study design is so critical. You can't just spray and pray.
Speaking of animals, this brings us to one of the trickiest rules in the document, the concept of biological relevance. I can't just grab a standard lab rat off the shelf because do cheat, can I?
You definitely cannot. This is arguably the most critical design element. The species you choose has to be biologically relevant.
Unpack that. What makes an animal relevant? Is it just about size?
No, it's about biology. Think of it like a software compatibility or a lock and key. If your viral vector is designed to unlock a specific human cell receptor to get inside, but the mouse doesn't have that receptor,
The key doesn't fit the lock.
the key doesn't fit. The virus won't get into the cells, so you inject the mouse, nothing happens, and you get a clean safety report.
Which is a false negative.
Yeah, completely. The data is useless because it doesn't predict what will happen in a human where the key does fit the lock. And if you're using a replication-competent virus, the animal model has to permit that replication. Otherwise, you're just drastically underestimating the potential viral load.
Okay, so we have the right animal. Now, what are we injecting into it? Does it have to be the exact vial I plan to give to patients? Because, manufacturing clinical-grade material is incredibly expensive.
It is expensive, but you can't cut corners here. The test article needs to be representative. Ideally, same manufacturing process, same titer, that's the concentration, and the same formulation as the intended clinical product.
Why does the manufacturing process matter so much if the genetic sequence is the same?
Because the process defines the impurities. If you change how you purify the virus, you might end up with more empty capsids, viral shells with no DNA inside. Those impurities can change how the immune system reacts, which in turn changes where the drug travels in the body. If your animal batch is cleaner or dirtier than your clinical batch, your map is wrong.
No research-grade shortcuts then. Okay, let's talk about the harvest. We've dosed the animals. What are we collecting? I looked at the list in section 4.6. And it is extensive.
It is a full body audit. The standard panel includes the injection site, obviously. Then you have the gonads. We will definitely come back to that.
I bet.
The adrenal gland, brain, spinal cord, liver, kidney, lung, heart, spleen, and blood.
That's the standard list. But the guideline says the panel can expand.
Right. It's not a static checklist. It has to be based on your vector. If your vector is known to target the eyes, you need to add eyes and optic nerves. If you're injecting into the spine, you need cerebrospinal fluid. You have to follow the biology.
Okay, we have our samples. We have a freezer full of tiny tissues. How do we actually check for the drug? What's the technology?
The gold standard, according to S12, is qPCR, quantitative PCR, or digital PCR. We are looking for the genetic sequence.
Wait, why aren't we looking for the protein? If I'm delivering a gene to make factor IX for hemophilia, shouldn't I be checking for the factor IX protein? That's what actually does the work.
You can, and you often should, using methods like ELISA. But, and this is key for the exam S12 mandates measuring the genetic material.
Why?
Think of it like a crime scene. The protein is the result of the crime, but the DNA is the fingerprint of the perpetrator. You need to know where the DNA went, even if it isn't making protein at that moment. The DNA could be lying dormant, posing a risk of integration, even if no protein is being made.
That's a crucial distinction. The DNA tells you about persistence and integration risk.
Yeah.
Okay, you flagged this earlier, and we need to drill down on it. The gonad rule, section 5.5. Why is there a specific call-out just for gonads?
Because the stakes are incredibly high. Biodistribution assessment in gonads, that's ovaries and testes, is mandatory for both sexes. The regulators are looking for the risk of inadvertent germline integration.
Germline integration. It sounds like a sci-fi nightmare. We were talking about passing the genetic modification to your kids, right?
Yeah.
Exactly. If the gene therapy integrates into the DNA of a sperm or egg cell, that change is permanent. It's heritable. It could be passed down to the next generation and the one after that. That crosses a major ethical line.
A line regulators are very nervous about.
Very. So if you find the product persisting in the gonads, it doesn't necessarily kill the drug, but it triggers a lot more work. You might have to do more studies to find out if the virus is actually inside the sperm or egg cells or just in the surrounding tissue.
Is there any way out of this requirement? Or is it absolute?
There is one main exception. You can skip one sex if the clinical indication is strictly sex-specific.
So if I'm treating prostate cancer,
You don't need to study ovaries. Or if it's uterine cancer, you don't need to study tests. But otherwise, you check both.
okay, that covers the core concepts. But the real world is messy. Let's walk through a few of the what-if scenarios. I feel like these are the essay questions on the exam.
Agreed. This is where the rubber meets the road.
Scenario 1, the unmatchable human vector. I have a gene therapy that targets a receptor that only exists in humans. No animal has it. I can't find a biologically relevant species. Do I just give up?
It's a tough spot, but S12 offers a path. You can consider alternative approaches. One common solution is to use a surrogate molecule.
A surrogate. A
Yeah.
stand-in.
Exactly. You manufacture a version of your product specifically for the animal. It has the same vector structure, same outer shell, but the inside or the binding site is tweaked to work in the mouse or monkey. It's like building a custom adapter.
So you build a mouse version of the drug just for the safety study.
Yes, or you could use transgenic animals, mice, that have been engineered to express the human receptor.
Okay. Scenario 2, the immune response pitfall. I'm using a common virus like AAV, but the monkeys I'm using have already been exposed to it in the wild. They have antibodies. What happens?
That will ruin your data. If they have antibodies, their immune system will attack the vector the second you inject it.
So it just gets cleared out immediately.
Right, and you won't get a true picture of where it would have gone in a patient without that pre-existing immunity.
So do I just suppress their immune system, give them steroids so they don't react?
No. The guideline explicitly warns against that. You should not immunosuppress animals just to get clean BD data. The solution is to screen the animals beforehand and only use the ones that are negative for antibodies.
Okay, scenario 3, contamination. This seems so practical, but how hard can it be to cut up a mouse without messing it up?
Surprisingly hard when you're looking for microscopic DNA traces with PCR. Imagine this. You inject the virus into the leg muscle. That injection site is hot. It has billions of viral particles.
Okay, it's glowing radioactive in this metaphor.
Right. Now, a technician cuts into that leg to take a sample. Then, using the same scalpel, they touch the liver.
Oh, no.
They just transferred thousands of particles. You run the PCR, the liver lights up, and you think your drug targets the liver when it doesn't. It was just a dirty scalpel.
A false positive.
Exactly. S12 requires a strict documented order of collection, usually from least expected concentration to highest, and changing your tools constantly.
One last practical question. I've done my study, I'm ready for the clinic, but then my manufacturing team says, hey, we changed the process. Or the clinical team says, we want to triple the dose. Do I have to redo the whole animal study?
The dreaded question. The answer is, it depends on the trigger. S12 lists specific triggers for new studies.
What are the big ones?
First, route of administration. If you switch from intravenous to, say, intrathecal into the spine, that is a massive change. You need new data.
Okay, what else?
Dose. If you significantly increase the dose beyond what you tested, you're in uncharted territory. You need new data. And if you change the vector serotype, like swapping AAV2 for AAV9, that definitely requires a new study. Their tissue tropism is completely different.
The key word there seems to be significant.
It is. Small tweaks might be justifiable with a paper analysis, but big shifts trigger a redo. And if you don't do it, the regulators will put your trial on clinical hold.
All right, we've covered a lot. Let's assume our listener is walking into the exam room in 10 minutes. They need the cheat sheet. What are the 7 essential takeaways they must memorize? Let's do this rapid fire.
Let's do it.
Point 1.
The definition. Remember the triad. Distribution, persistence, and clearance. Target, AND, non-target tissues.
Point 2.
Timing must be completed before clinical trials, no exceptions.
Point 3.
Biological relevance. The animal species must be permissive. If the key doesn't fit the lock, the data is garbage.
Point 4.
Methodology. qPCR is the standard for measuring the genetic material, not the protein.
Point 5, the big one.
The gonad rule. Analyze gonadal tissue in both sexes for germline risk. The only exception is a sex-specific disease.
Point 6.
Representative material. The test article must match the clinical product. No research-grade shortcuts.
And finally, point 7.
Triggers. Know what forces a redo. Big changes to the route of administration, the vector, or the dose.
That is the list. If you have those 7 points locked in, you are in good shape.
I would add one final thought for the big picture. The guideline does touch on the future. As we gather more data on these vectors, like AAVs, we're starting to see patterns. The hope is that we might eventually be able to rely on platform data.
What does that look like?
Well, if we know exactly where AAV9 goes in a monkey because it's been tested hundreds of times, maybe we don't need to run a new animal study for every single new payload we put inside it. We could rely on historical data.
That would be a huge win for the 3 Rs for reducing animal use.
Exactly. That is the ultimate goal. But we are not there yet. For now, we follow S12 to the letter.
All right. One last tip for me. Before you close your books, open the PDF and look at section 4.6. Just read that list of tissues one more time. It's the kind of detail that separates an A from a B.
Good advice.
Good luck on the exam. You've got this.
Good luck.